We know a fraction of cancers develop as a result of known genetic mutations. For example, approximately 10% of breast cancers come from known genetic mutations like the BRCA1 and BRCA2 genes. But what about the other 90%? This morning, how better understanding our genetic code may someday transform the way we identify and treat those cancers without a known genetic mutation. Bench to Bedside begins now.
From the research bench to the patient's bedside, this is Bench to Bedside with your host, Vice Chancellor and Director of the University of Kansas Cancer Center, Dr. Roy Jensen.
Good morning. I'm Dr. Roy Jensen, Vice Chancellor and Director of the University of Kansas Cancer Center. Joining me this morning to talk about how understanding our genetic makeup may help us develop more precise therapies is Dr. Jennifer Gerton, an investigator at the Stowers Institute for Medical Research, and Mary Frances Haake, a patient research advocate. Good morning to the both of you. Mary Frances, I'm going to start with you. You are a member of PIVOT, which is the Cancer Center's Patient Research Advocacy Network. And advocates like yourself give our scientists the patient perspective. And the result, obviously, is a more patient-centered approach to research. Tell us a little bit about how you became involved in PIVOT.
Thank you, Dr. Jensen. I'm happy to. I attended the patient advocacy training through PIVOT in January 2020. I had supported many friends in their breast cancer journeys, and I believed that advocacy training would benefit me, my friends, and the providers. So after that Saturday training, I then attended the monthly PIVOT meetings and some rapid reactor team meetings as well.
I see. So Dr. Gerton, tell us a little bit about what you study in your lab.
Yeah, so my lab studies, as a general topic, we study something called genome integrity. And so the human genome is made up of 23 pairs of chromosomes. And every time a cell divides, those chromosomes have to be transmitted from cell to cell because they really contain the information that's the blueprint of life. And so what we really study is how those 23 chromosomes get passed from cell to cell to cell. And we know that sometimes in cancer, that process isn't working correctly. So if we understand that basic process, then we might understand something about cancer mechanisms.
Mm-hmm. So I've heard you speak a number of times, and I know you have a fantastic analogy that helps kind of explain the relationships between basic science, clinical science, and patient care. Could you tell our audience about that analogy?
Sure, I'd be happy to. So I like to think about the biomedical research enterprise as a tree. And if you think about a tree, it's got, leaves and branches, but it's also got roots and a strong trunk to have a really healthy tree. And so when in healthcare, the things that we want are things like diagnostics and medicines and various therapies. And in order to have those, I think of those as like the terminal branches and the leaves of the tree. But in order to keep producing these really useful, healthcare related products, we have to have these healthy roots. And the roots and the trunk to me are like the basic science research. We have to have this foundation to understand how the cells in our body work in order to produce these things that are useful to us.
Mm-hmm. That analogy never gets old, Dr. Gerton. And I want to turn now to Mary Frances. How did you specifically get connected with Dr. Gerton after you came on to PIVOT?
The outreach and engagement program for basic scientists, and I signed up to participate immediately due primarily to my experience with the rapid reactor teams. I really appreciate the collaboration there between advocates, clinicians, and scientists. So Dr. Gerton and I were assigned to each other by staff, and once partnered, we took part in some monthly training, We received communication and leadership coaching as well as team building. Then the Pivot staff suggested that we get together on our own to develop a relationship. And on our very first Saturday walk, Dr. Gerton shared her scientific research and I shared my patient experience. And that perspective really enlarged our perspectives. Then we began working on our scientific presentation based on Dr. Gerton's research, and we found more that we had in common between her work and my patient experience.
I see. So Dr. Gerton, why is it important to know if a genetic mutation, a patient's germline, DNA, is driving a person's cancer?
Yeah, so I'm not a clinician, but what I understand is that kind of information really guides the healthcare decisions that get made. So if someone comes in and has a mutation, the doctor that's working with that patient might decide on a specific medicine that they think might be more impactful for that specific mutation. And then it's also going to impact things long term. So how often does that person need to be screened for other cancers? And how is that going to impact actually the family of that person? So we need to think about things like if someone carries a mutation, have they passed that on to their children, and how is that going to impact their health care? So I think it really can make a big difference if we know what the genetic cause is of a cancer.
Mm-hmm. So obviously breast cancer and hereditary breast cancer gets a lot of press, and my laboratory actually happens to work on BRCA1. But genetic differences are very important in a wide range of different types of cancer. Could you tell us a little bit more about that, Dr. Gerton?
Sure. So I think there's this whole field of cancer genetics, which you're, of course, extremely well versed in, where there's just a whole bunch of researchers that are trying to understand these genetic causes of cancer. And for many cancers, like breast cancer, we don't know for a large fraction of them what the genetic basis is, or even if there is a genetic basis. And so that's where this sort of foundational research comes in. We want to be discovering new kinds of genetic contributions that we can then someday, many years down the road, may really impact the way that we can more successfully approach these healthcare outcomes.
Mm-hmm. So if you're just joining us, we're here with Dr. Jennifer Gerton, an investigator at the Stowers Institute for Medical Research and an investigator in the University of Kansas Cancer Center, and Mary Frances Haake, a breast cancer survivor and member of Pivot. Remember to share this link with people you think might benefit from our discussion. Use the hashtag #BenchToBedside. Now I want to go back to your work, Dr. Gerton. You are studying a very specific part of the chromosome, which is called the centromere. And Mary Frances, you have a great analogy of the centromere's role in the function of chromosomes and the DNA sequence. Can you share that with us?
I'd be happy to. And this is the result of my partnership. This understanding is a result of my partnership with Dr. Gerton. So, what we know is the centromere is a specialized DNA sequence of a chromosome, and it has two structural roles. First, the centromere links a pair of sister chromatids, which are a copied chromosome pair, in a process called cohesion. And so the first metaphor is I think of the centromere in this role as a cummerbund, holding those two sister chromatids together at their waists. The second job of the centromere takes place when the cells split, which is called mitosis. The spindle fibers attach to the centromere by microtubules through the connector core complex, which physically splits the pair of chromatids apart. And here comes my second metaphor. The centromere and the connector core make a carabiner, that metal ring we use in mountain climbing. And the microtubules are the ropes that attach to the centromere. So together, they physically split that sister chromatid in two.
That's a great analogy. So, in terms of your own research, what happens if this particular region of the chromosome is not doing its job, Dr. Gerton?
Right. So as I mentioned earlier, we're really interested in genome integrity. And so this part of the chromosome is really important for taking that set of chromosomes from cell to cell to cell every time the cell divides. And so if the centromere isn't working, one of those chromosomes might not get carried into the next cell, and that's going to result now in an altered blueprint. And that really could be a foundational problem for something like cancer.
Mm-hmm. So as you mentioned in your earlier analogy, basic research functions as the roots or the foundation of our understanding of cancer at a molecular and biological level. And every advance that we've made in treating cancer over the last few decades has really started with basic research. How do you think that the research going on in your laboratory can potentially lead to better treatments for cancer or other types of diseases?
Right. So I think in order to have, like I said, have these things like diagnostics, genetic tests, in order to have things like personalized medicine, treatments that are really tailored to a particular cancer, we need to understand the basics. And so that's why we really want to understand these centromere regions. They're not an area that's really been explored as a potential way that a person might be predisposed to cancer, but they are inherited from your parents. And so we really need to understand just their basic biology more so that maybe 20 years from now we can think about how we might use them, whether that's to select a better therapy, or whether that's to help with the prognosis of disease, or to think about the inheritance within a family.
Mm-hmm. So one of the reasons we wanted to have you on the show this morning, Dr. Gerton, is that there was a recent announcement that scientists had finally completed the sequencing of the human genome. And I think a lot of people had assumed that had been done. Mm-hmm. But a little known fact between folks like you and me was that it hadn't quite completed the sequence, but you played an important role in really getting us to that completed sequence of the human genome. And tell us a little bit about why that project was so important to finally finish that Herculean effort.
Right. So I first just want to kind of let people know that it's really interesting. This project, which was a consortium of 100 scientists spread across the world, really happened during the pandemic. So it was really, for a lot of us, when our labs were shut down, it was really a bright spot, something we could look forward to, working with this very large group of people who were very motivated towards this common goal. So it was just really a delightful professional experience. And there was about 8% of the genome where we had never had sequence information before, DNA sequence information. And so when people do their experiments, a lot of times what they do is they're using a common reference genome. Okay, but the genome has been missing certain parts, including these centromere regions that my lab is so interested in, and other parts of the chromosomes too. And so by filling in this 8% of the genome, it really allows those parts of the chromosomes to become part of this scientific discovery process, where they've really we've not been able to study their biology very effectively before. So for us, it's really going to change the way we can do research in the laboratory.
Yeah. So Mary Frances has already told us about your walks together. Mm-hmm. Could you tell us a little bit about how the partnership between your lab and Mary Frances has really influenced the research that's going on in your lab, Dr. Gerton?
Yeah, so I'm a basic scientist, and Mary Frances had this cancer experience, and so what by her sharing this with me, it really helped put my research into context of how someday it might help a patient. So she shared two really personal things with me. One was she actually shared her document when she received her genetic diagnosis, and it turned out she didn't have these BRCA mutations, and so we don't really know what the genetic origin of her cancer was, and that kind of allowed us to have this interesting conversation. Well, you know, could it be she has a centromere that was predisposing her to get cancer? The other part of the personal connection was that we're both moms and we love to talk about our kids. And so we really talked through this piece, like what would it look like if she had received a genetic diagnosis, and how would that impact her kids and her daughter and her long-term health care. So it was really kind of this personal connection that helped me think about the research that we do in the lab, how it could have this, you know, many years from now have an impact on that experience.
Mm-hmm. So I want to switch gears back to you, Mary Frances. You go to the Cancer Center's Breast Cancer Survivorship Clinic. Could you tell us a little bit about your experience with this clinic?
Absolutely, I'm so happy to. So I had my original diagnosis July 8th, 2010. And once I was released by my medical oncologist, I wanted to continue my care in breast survivorship. I wanted an annual touch point. I wanted blood work. I wanted to be examined once a year because there's so many medical innovations taking place each year, and I wanted to take advantage of that. Lori Rinaldo is my provider, and she's the sole provider in breast survivorship, and she's cared for patients for more than 25 years. My primary care doctor is also at KU, so they can work together if I have a health issue that's affected by me having breast cancer or separate from that. I have had some health complications since that diagnosis, and my provider worked closely with Lori because of Lori's understanding of the benefits and risks of treatment for other conditions based on breast cancer survivorship. What I found really interesting that I just learned is that breast survivorship has become an endowed chair at the KU Cancer Center, which means that nurse practitioners, physician assistants, and physicians can receive focused training in breast cancer survivorship care. And thinking that, 1 in 5 women will have breast cancer, I'm really encouraged that other providers are interested in learning how to provide that care.
All right, thank you. So we're approaching the end of today's episode, and I wanted to mention for our viewers who may be interested in joining or learning more about Pivot, you can email pivot@kumc.edu or visit kucancercenter.org/pivot. One more question for each of you. Dr. Gerton, what would you say excites you most about the future of genetics research?
Yeah, so I mean, as part of this telomere to telomere consortium, I'm really excited about this new 8% of the genome that we haven't had access to before. And I think the next 10 years is really going to see us laying, like I said, this basic science foundation for how these parts of the chromosomes work and the basic biology of the genome. And then hopefully, 20 years from now, we can take that information and use it to help healthcare decisions.
Mm-hmm. And a follow-up to that, which I hope we're not going to get too far in the weeds here, but if this section of the genome was so hard to sequence initially, does that mean that the sequences of tumor DNAs that we have in our databases, does that mean that it's not covered and we need to do something extra to look for mutations in those regions?
That's a great question. So because that part of the genome was so difficult to assemble, it took a lot of new technologies in order to assemble it for one genome. The kinds of data that we already have on these regions are not going to be amenable to those very specialized approaches that we had to take. But that information is there, and so what our job is now is to find creative ways to use it to understand better what happens in cancer, whether there are mutations there. And I just want to say one other thing about centromeres, which is that they're this incredible signature in each person's genome. And so each person has a very different set of centromeres. And so we're very curious to just see how this human variation plays out in terms of centromere function. And again, we can really take advantage of all of the sequencing information that's there, but we have to come up with new computer programs and algorithms to analyze it.
Sounds like a lot of exciting work going to be going on in the Gerton lab. Well, very good. So, Mary Frances, tell us a little bit about what you feel like you have gained in helping other patients like yourself.
Well, I feel like Pivot has helped me gain skills. You know, before I had empathy and care, I think now I can help translate medical jargon for women participating in their care. I can also ask questions that they may be uncomfortable in asking. I really have a lot of hope because not only of the innovations, but just because of the experience that I see of the care that patients get, you know, in the clinics, whether it's, you know, with the providers, the medical assistants, or the staff greeting them. So I get a lot of hope and gratitude for my own health.
That's fantastic. Well, thank you so much. Thank you, Dr. Gerton. Thank you, Mary Frances, for being part of this week's episode of Bench to Bedside. To learn more about our basic research program and what we are doing to better understand where cancers come from, visit kucancercenter.org/cancer biology. That's it for today. Join us next week at 10 a.m. for Bench to Bedside. Thanks for watching. Subscribe to our Bench to Bedside podcast, now everywhere podcasts are available.